Picture yourself standing in a humid tropical forest somewhere between India and the Solomon Islands. This is where our story begins. Thousands of years ago, wild banana plants grew here, naturally producing fruit filled with hard seeds. Two main species dominated these landscapes: Musa acuminata and Musa balbisiana. These wild ancestors were nothing like the bananas we know today. Early hunter-gatherers walking through these forests would have encountered these plants. The fruit was edible, but eating around dozens of seeds was hardly enjoyable. Yet somewhere along the way someone noticed something unusual. A few rare plants produced fruit without any seeds at all. This remarkable trait is called parthenocarpy. It means the plant can develop fruit without pollination occurring. No pollination meant no seeds forming inside for early foragers. This was a delicious discovery. But here was the problem: seedless bananas cannot reproduce on their own. They cannot spread their seeds, because they have none. The first banana farmers solved this puzzle. Through careful observation, they realized that banana plants grow new shoots from their base. These offshoots, called suckers, could be dug up and replanted elsewhere. By transplanting these shoots from the seedless plants early, farmers could propagate the exact same desirable fruit. This was the birth of banana cultivation. Archaeological evidence from Papua New Guinea pushes this story back at least 7,000 years. Ancient drainage systems and cultivation remains suggest that banana farming was already well established by then. These were not casual experiments, but sustained agricultural practices. As humans migrated and traded across the region, they carried their prized banana plants with them. Different subspecies that had evolved separately for millennia now crossed paths. This mixing created new hybrid varieties with combinations of traits their wild parents never could have produced alone. Later, banana cultivation reached Africa, where it became a secondary center of diversification. African farmers developed distinct varieties adapted to their local conditions. Plantains and East African highland bananas emerged from this independent experimentation, becoming crucial food sources for millions. Let us talk about what makes your supermarket banana so different from almost any other fruit you eat. It all comes down to chromosomes. Most plants and animals have two sets of chromosomes, one from each parent. Scientists call this diploid. But the bananas you eat have three sets. This is called triploidy, and it is the secret behind everything that makes modern bananas so convenient. Think of it like this: a normal diploid plant gets one chromosome set from mom and one from dad. Easy math. But triploid bananas break the rules during reproduction. Something peculiar happens called meiotic restitution. One parent accidentally contributes both copies of its genome. Instead of just one, the other parent contributes, normally providing a single copy. Add those together, and you get three. Now here is where it gets really interesting. Scientists classify bananas using a simple letter code based on which wild ancestors contributed those chromosomes. The letter A represents Musa acuminata, while B stands for Musa balbisiana. Your typical yellow banana. The Cavendish is AAA. That means all three chromosome sets came from acuminata ancestors. The famous Gros Michel, which dominated global trade before the 1950s, is also AAA. But plantains, those starchy cooking bananas essential to millions of people in Africa and elsewhere, are OB. 2 doses of acuminata, 1 of balbisiana. Then there are the AB varieties, which tilt the balance toward balbisiana genetics. This triploid condition comes with an unexpected bonus for anyone who loves eating bananas. Having three chromosome sets messes up meiosis completely. The chromosomes cannot pair up properly during reproduction, so the plant cannot make viable seeds. That seedless convenience you enjoy is actually a biological accident. A happy mistake of chromosome mathematics. Between these fully triploid varieties exist intermediate diploid stages. All bananas have two acuminata genomes, while AB types carry one of each ancestor. These represent evolutionary stepping stones on the path to the three-set formula that changed banana history forever. Now here, where the story takes an unexpected turn beyond just having extra chromosome sets. Scientists discovered something hiding in the genetic architecture of cultivated bananas using sophisticated techniques called mate pair sequencing and backfish analysis. Researchers uncovered large-scale chromosomal rearrangements that had gone unnoticed for centuries. Here, what they found in a particular wild banana subspecies called Malaxensis, two chromosomes had essentially swapped pieces of themselves, a segment of about 3 million base pairs from the end of chromosome 1, exchanged places with a segment of about 10 million base pairs from chromosome 4. This is what scientists call a reciprocal translocation. Now you might think this kind of rearrangement would be purely destructive, but in the banana world, it turned out to be surprisingly useful. When these rearranged chromosomes try to pair up during reproduction, they create what researchers call segregation distortion. The chromosomes simply cannot align properly, which leads to dramatically reduced fertility for wild bananas trying to reproduce naturally. This would be a serious disadvantage, but for early farmers selecting plants with fewer seeds, this chromosomal quirk was essentially a gift. The translocation creates a genetic link between chromosomes 1 and 4, tying them together in ways that reduce recombination and promote sterility. The evidence for this is striking. When scientists surveyed banana varieties, they found this rearranged chromosome structure appearing frequently in triploid cultivars, yet it remains rare. In truly wild banana populations, this pattern tells us something remarkable. The very genetic change that makes bananas less fertile may have been actively selected by early farmers who had no idea about chromosomes or translocations. They just knew which plants gave them the seedless fruit they wanted. Nature provided the mutation, and human preference did the rest. Scientists have finally cracked the genetic code of our most beloved banana varieties by sequencing the complete genomes of both Cavendish and Gros Michel at the chromosome level. Researchers assembled an enormous picture. The Cavendish genome spans 1.48 gigabases, while Gros Michel comes in at 1.33 gigabases. But the real discovery came when they looked deeper. Hidden within these triploid genomes were 3 distinct subgenomes, each traceable to a specific wild ancestor. Scientists named them BAN DH and Z. These are not random labels, they are genetic signatures pointing directly to the wild subspecies that contributed each set of chromosomes. BAN maps back to Musa acuminata, subspecies. Banksii DH traces to Malaxensis, and Z leads straight to Zebrina. Here, what makes this so remarkable? These 3 wild subspecies never naturally encountered each other. Banksii grew in New Guinea and surrounding islands. Malaxensis thrived on the Malay Peninsula. Zebrina was isolated in Java and the Indonesian archipelago. They were separated by hundreds of kilometers of open ocean and dense rainforests. So how did their genes end up in the same fruit? The answer lies in human movement during the Holocene epoch. As human populations expanded and migrated across Southeast Asia and into the Pacific, people carried banana plants with them. Suckers and corms traveled in canoes, moved along trade routes, and were replanted in new settlements. This human-mediated transport bridged geographic barriers that had kept these subspecies apart for millennia when these previously isolated Subspecies finally met in cultivated gardens, hybridization became possible. The genetic evidence now confirms what botanists had long suspected. Your breakfast banana is essentially a living record of ancient human migrations, carrying the genetic fingerprints of 3 distinct wild populations that humans brought together. Scientists have finally cracked the genetic code of our most beloved banana. Varieties by sequencing the complete genomes of both Cavendish and Gros Michel at the chromosome level, researchers assembled an enormous picture. The Cavendish genome spans 1.48 gigabases, while Gros Michel comes in at 1.33 gigabases. But the real discovery came when they looked deeper. Hidden within these triploid genomes were 3 distinct subgenomes, each traceable to a specific wild ancestor. Scientists named them BAN, DH, and Z. These are not random labels. They are genetic signatures pointing directly to the wild subspecies that contributed each set of chromosomes. BAN maps back to Musa acuminata. Subspecies Banksii DH traces to Malaxensis, and Z leads straight to Zebrina here. What makes this so remarkable? These 3 wild subspecies never naturally encountered each other. Banksii grew in New Guinea and surrounding islands. Malaxensis thrived on the Malay Peninsula. Zebrina was isolated in Java and the Indonesian archipelago. They were separated by hundreds of kilometers of open ocean and dense rainforests. So how did their genes end up in the same fruit? The answer lies in human movement during the Holocene epoch, as human populations expanded and migrated across Southeast Asia and into the Pacific. People carried banana plants with them. Suckers and corms traveled in canoes, moved along trade routes, and were replanted in new settlements. This human-mediated transport bridged geographic barriers that had kept these subspecies apart for millennia. When these previously isolated subspecies finally met in cultivated gardens, hybridization became possible. The genetic evidence now confirms what botanists had long suspected. Your breakfast banana is essentially a living record of ancient human migrations, carrying the genetic fingerprints of 3 distinct wild populations that humans brought together. Let me take you back to the early 20th century. There was a banana that most people had never heard of called Gros Michel. It was the superstar of the banana world, thicker skin, richer flavor, and it shipped beautifully across oceans. If you ate a banana before the 1950s, this was almost certainly what you were eating. But then disaster struck. A fungus called Fusarium oxysporum. Specifically, race 1 began spreading through banana plantations like wildfire. This pathogen causes what farmers call Panama disease. It clogs the plant's water vessels and slowly chokes it from within. By the 1960s, Gros Michel had been virtually wiped out from commercial production. The banana industry's solution was Cavendish. This variety happened to be resistant to race 1 of the fungus. It became the new global standard. The banana you see in grocery stores today, problem solved or so. Everyone thought. Now, history is repeating itself in a terrifying way. A new strain called tropical race 4 has emerged, and this one attacks Cavendish bananas with devastating efficiency. It has already spread through Asia, Africa, and the Middle East. Scientists are racing to understand why this is happening at the genetic level. The answer lies in the DNA of these 2 varieties. Researchers have discovered that Gros Michel lost a crucial genetic region called the RLP locus in one of its subgenomes. This loss stripped away its ability to resist race 1 of the fungus. That is why it was so vulnerable. But here is the sobering reality: both Gros Michel and Cavendish share a different problem. They both have repetitive DNA sequences jammed into the promoter region of a gene called RGA2. These insertions act like roadblocks, preventing the gene from producing enough protein to fight off tropical race 4. In other words, the very genetic feature that could save Cavendish from the current threat is essentially switched off. We are watching the same vulnerability play out twice in a single century, and this time there is no obvious replacement waiting in the wings. Now let me tell you about something remarkable that scientists discovered hiding inside banana fruit. They found two special genes called NAP homologs. These are transcription factors, which is just a fancy way of saying they are genetic switches that control other genes. What makes these NAP genes so interesting is where they show up. They are specifically and highly active in fruit tissue, which immediately told researchers these genes must be important for the fruit itself. But the real breakthrough came when scientists used a technique called ChIP-seq to see what these NAP genes were actually doing. Think of ChIP-seq as a molecular detective tool that reveals where proteins bind to DNA. What they found was striking. These NAP transcription factors were binding directly to the control regions of dozens of different fruit ripening genes. In other words, these two NAP genes act like master switches. When they turn on, they activate an entire cascade of other genes that control how the fruit ripens. This discovery is a big deal for banana breeding. For a long time, banana improvement has been slow and difficult because scientists lacked a clear map of which genes control which traits. Now, with these high-quality genome assemblies and the identification of these master regulator genes, researchers have a powerful new platform. They can identify candidate genes for fruit quality traits much more easily. They can work on molecular breeding approaches that were simply not possible before. The researchers call this super domestication. It means using genetic knowledge to rapidly improve crops in ways that traditional breeding could never achieve. With these tools, scientists can now work toward bananas with better flavor, longer shelf life, and improved disease resistance. The ripening code has been cracked, and it opens doors to a future where bananas can be precisely improved rather than just hoping for the best.